If you're into plants, the idea of creating your own hybrid has most definitely crossed your mind. But when it comes down to Alocasias, they are notoriously tricky to hybridize. So did I actually manage to create my own hybrid? Well, kinda. So come with me as I take you through the journey of Alocasia pollination and how you can create your own hybrid, and then the story of what happened with mine. Hey everyone, Pablo from Aroid Vibes Only, and in this video I'll be creating the video I wish I had when I had started my journey with Alocasia. When I first started my Alocasia journey, there were a lot of posts that I found that were kind of confusing when it came down to Alocasia inflorescence or flowers. Some of them were talking about always cutting them, always about never, when you should, like how do you collect pollen, how do you even begin to put that pollen at the right time into the inflorescence, like how does that work? I was just looking for answers, and honestly, it was difficult to find. It was very confusing. There's a few blog posts here, there's a few things on Instagram, but there was not one video that completely told me everything that I wanted to know. And that is the reason that I'm making this video. I'm making it for you, and I'm making it for me because I wish I would have had this video. So Alocasia hybrids are nothing new. First of all, they happen naturally in nature. Whenever the territory between two different species kind of overlaps, they will naturally just create hybrids. Very, very common. It happens always. This is what drives speciation and then also just like evolution as well. But when it comes down to humans artificially creating them, it has also been around for a couple hundred years. Actually, a lot of the hybrids that are most popular come from like the late 1800s from the Gentry brothers, I believe. Like I'll correct myself in the screen what their name was. And they created a lot of the different hybrids that kind of were really popular at the time.
And of course, if we're going to be talking about hybrids and the 1800s, we need to mention the Veitch family. They were a dynasty of plant botanists who set out around the world to collect specimens from all the countries. They had an entire army of botanists and collectors that would just send the plants back to the UK. They basically had a control of a lot of the new hybrids and plants from the early 1800s until the mid 1900s. They introduced over 1,000 species plants into cultivation. By many, they're considered pioneers of botany, and our current plant selections could not exist by them. By others, they are considered the embodiment of British colonialism, in which they were able to go around the world, poach all these specimens from the wilderness, collect them with locals, and then bring them back to the UK by any means necessary. Personally, I think it's important whenever we're talking about something that happened in the past to be able to look at it from their perspective and not just from ours, coming from a modern ethics and understanding. Like, yes, this is poaching, but back then this would take years by boat. They were unsuccessful, and they really pioneered the hybridization in orchids and a lot of other species, and species that are just part of everyone's gardens nowadays. So regardless how you feel or we can feel about them now, it is always important to know about them and the impact that they had in botany across the board.
So you get a cohort of famous hybrids from that era, and then you kind of go through this black period up until like the late 70s. I don't know what happened in the first half of the 20th century, but I did not get a lot of information of people. Oh, well, I guess I do know what happened, but the information of plants and hybrids and all that kind of just gaps until the late 80s and then 70s, and that's when you begin to get a lot of those outdoor Alocasia kind of hybrids like Calidora and all of that stuff kind of picking up, and eventually you end up with some of the most famous ones now like Maharani I would consider to be some of the more known and popular types of specific kind of hybrids. Some of the challenges that come with hybridizing Alocasias have to do with the pollen availability and the window to use it. So Alocasia pollen is really delicate and frail. I'm trying to figure out a way of making it sort of stronger or activating it early. So if you're interested, at the end of this video, I'm going to just go through like some of my ideas of what I'm trying to figure out and some overall questions that I just have as I research and learn more about them. So what happens is storing the pollen is difficult, the viability of the pollen is difficult. So many times the people that end up with actual hybrids are people like myself who have a lot of plants and end up having multiple inflorescences kind of overlap. So at the same time that I have these 4 ones here, my silver dragon has another 4. I can see back here my pink dragon has 2. My purpley has one. So you begin to see all of the different options around you. So the hybrid that I basically created, and the idea that I had was honestly just to kind of get a system down. Like my goal is to standardize and create a way of all of us to just be able to create whatever hybrids we want. Like it cannot be that rocket sciencey. So I gave it a whirl, and what I had available at the time was Alocasia Corazon. Corazon to me is one of the most kind of like stud like type of plants. What's the word, like yeah, I mean, it just produces so much pollen. Here you can see one of the inflows that have aged a little bit is absolutely like covered in pollen. Like, oh, you see all of that around it? Yep, that's all pollen. Underneath this leaf it's full of like pollen because it keeps falling in there. So Alocasia Corazon to me has been a great sort of source of fresh pollen because it produces a lot and I'm able to store it and just kind of play with it. So that was the pollen donor, and the ovary, or where the actual inflow was coming out of, was this plant. This is my Alocasia longiloba variegated. The type of variegation that this plant displays looks a lot better in pictures, like video just really struggles. And it tends to be a more type of like marbled, light green, yellowy. Sometimes it will burn in more yellow as time goes on, but overall this plant has more of a mottled kind of dark green and light green. And then the good thing is all of the corms that come from this plant are basically variegated. So all of them come out with the same characteristics of burning green, sort of coming out light green, and then they kind of burn yellow, and the other parts burn deeper green. So we have two mature plants that are both producing inflorescences, and they're ready to work with each other and create some kind of fun hybrid. But before we do that, I want to take a step back with you and go over all of the anatomy kind of parts of an inflorescence. I think this is going to be very important because that way you're going to kind of understand why the timing is important to when you use the pollen, and it also going to make sense why it's so difficult to get this done, and also why they put out so many inflows. While personally I don't consider it incorrect if you say Alocasia flower or you call it a flower, like I don't care, I understand what you're meaning. The correct term for an Alocasia flower is an inflorescence.
Whenever you think of a flower, you think of something like this. This is considered a perfect flower because it has both the female and the male components in the same flower. But you should think of it more like a single flower, and in nature there's a lot of diversity. So single flowers come sometimes as a pack. Whenever you have a lot of them together, it's called an inflorescence. And then the different arrangements have different categories, but the two that you should know about are going to be the cymose or determinate. This is whenever the bloom or the inflorescence reaches the top flower, the entire structure dies. The one that you're probably the most familiar with is going to be the death bloom in agaves, which it creates a giant stalk and you have flowers along the stalk until it reaches the top one. And when the top flower opens, the entire structure dies. The second one is going to be racemose or indeterminate. This means that whenever the inflorescence comes out and it begins to mature, as it's maturing, other inflorescences can come out and mature at different types, so you have an ongoing number of inflows and fruit. What you can think of, and it'll be super easy to remember, is going to be grapes, because grapes come out in bunches and these different bunches are maturing at different times. So whenever it happens, it is not determinate, it's indeterminate. And if you speak Spanish, it's going to be super simple to remember the name, because in Spanish, bunch is a racimo de uvas, and racimo has the same Latin root as racimos, which is racemus. And the one type of indeterminate inflorescence we care about is this one. This is called a spadix inflorescence, and it is shared among currently all of the known aroids, but I'm sure there might be an exception hidden somewhere. And the one that we care about is, of course, is going to be the Alocasia inflorescence. So from the bottom, you're going to have first the female zone, which is going to have something that looks kind of like berries. They are called pistillate flowers. It is a pistillate because it is separate from the male area. Then you're going to have a sterile section. This is basically an area that is there to prevent the self-pollination of the plant. So it's trying to separate the male zone from the female zone. So above the sterile interstice, you're going to have the male zone, and basically the pollen comes out of here around the synandria, which is just those little hexagon structures on top, and it kind of oozes out of that in this kind of semi-dry way. And above that, you're going to have the appendix. The appendix is considered to be needed to change temperature. It helps the smell kind of go a little further, and it is considered to be key for the pollination and attracting the pollinators in all of the aroids, particularly like Amorphophallus. So then you have a structure that covers and encases the entire thing that is called a spathe. That is what kind of folds over, stands up, and kind of gets crunchy afterwards. And then all of this put together, the female, the male, and the appendix is considered the spadix. So this is a spadix type of inflorescence because it has this type of arrangement. Something that is important to note is the timing. So because you have this female and male zones, they do not activate at the same time. The first thing that happens is the bottom part is going to activate. The female zone will become active, start smelling, and this causes the spathe to open. So it creates almost like a cup around the appendix. It almost guides everything. It's almost like a baseball glove. So everything that it catches, it sends down towards where the female area is. Then you're going to go through a very short window, which is around 24 hours. And then that interstice, that sterile area, closes upon itself. That is why the flower almost seems like it closes. And then once it has closed, that is whenever you get the pollen to come out. So you're going to have the female area being receptive first. You're going to have the separation, it closes, and then you have the pollen coming out.
So now that you're an expert in the inflorescence anatomy in plants and we understand all the different multiple parts, let's talk about the timing and why it matters. So when you think about it logically, the plants are going out of their way to not self-fertilize, right? They have specific times when they're fertile and they have that smell and it's kind of viable. And then the rest of the time it locks, like it closes, it produces the pollen, and then it really requires the pollinator to sort of like be on top of it. As we talked, the pollen viability is kind of frail. So it makes a lot of sense for you to have 4, 5, 6 flowers coming out at different times and all of them maturing at different sort of stages. That way you increase the odds of you actually getting some kind of pollen from something else. So if you have an insect going around different inflows, you will most likely end up with some DNA genetic material that is not yours. So that is the reason why they go in this kind of quantity amount rather than quality, because they know most likely many of the inflows will fail, but they need to sync up to other plants near it in order to succeed. So some of the different stages that you will find in your inflows, I consider it to be 4 main stages. And the second stage is literally the one that is when you should be doing the pollination. So this is what I consider to be like the first kind of phase. This is the same way that they come out of like the stem and is like the original. Right now you have the spadix, everything around it completely closed, completely sealed. This plant is what I would consider to be like green. It's still not mature enough to be viable, so you just need to give it a little bit more time. The second phase, I do not have any of it yet because it lasts very, very little. But that is whenever you get the spathe, like this is like the spathe around the entire thing, when it's like first opens and is like standing up. So basically you end up with a plant that is opening the spathe. This is when you should be ready to put your pollen. So we'll open the spathe and then after a few hours, it begins to create a smell. I find that different alocasias have different smells when it comes down to when they're ready to be viable. Corazon is one of the most stinkiest dragon breath, also super stinky. Maharani, I can literally smell when I'm sleeping and I know I'm gonna wake up to an open inflow. Some of them smell super strong. Other ones like the Reversa, it has very little smell. I never kind of smell it. Other ones like the pink dragon, some people describe as sweet smell. Personally, I don't see that. Most of the time, to me, it smells like gasoline. It smells, it has that kind of gasoline-y, petroleum-y, kind of chemical kind of smell to it. Eventually, you get used to it. I mean, I have so many plants, and all of them are putting so many inflows that I literally breathe in that smell. So I'm more than used to it. But if you have never gotten one when it opens and it smells really bad to you, that is a normal process, and it's actually telling you that you should be getting your pollen ready. So that is whenever you should be opening the sides of this sort of this paddocks. So you're gonna go all the way down to the female zone and kind of like open the windows on the side. So whenever you're getting that smell, that is the second phase at its peak, and whenever you should be getting ready to give the pollen, because that is when it's the most optimal time. Then the next one is going to be like this one. Whenever you get the actual spathe to fall back, you still might have the appendix still standing up, still looking fresh. That doesn't matter. In my experience, the moment the spathe comes down, it's done. Like that is when your window closed. That's that whenever that sterile interstice kind of closes to prevent self-fertilization and leave the pollen out. And also I think it does it to kind of force the insects to go through. So that is the third phase. That means that it's too late. And then, so the fourth and final stage are two possibilities. The first one is a failure. So this is whenever the inflow just shrivels up and it just becomes like super dry. It becomes nothing in it and it's basically done. That means that it's a failed inflorescence. And you will know fairly quickly depending on the species. So there's some species that will fake you out because the berries that it have, like they look like they were fertilized, but they're not. For example, Maharani, even though it's considered sterile, like if you try to play with it and kind of fertilize it and put some pollen, it's going to stay kind of green for a long time and eventually just die. Aslanii, also known for keeping, like, at least with me, like those berries stay on for like 1 month, 2 months, and then it just aborts it. And I'm like, damn, so it was just a lie, all a ruse. But usually most of them, for example, Corazon is really, really good at being like not fertilized, dead. If you're feeling the stem and it's kind of dry and you're feeling this, that's it. That your stem is done. Your entire journey was a failure and you get to try all over again. And the other possibility is that you are successful and you end up with something that is called an infructescence. What that means is that you're starting to create a fruit. In this case, I believe they're considered berries because they are true berries. So now that you're familiar with all of the anatomy parts of the flower, the inflorescence of Alocasia, and you're also familiar with the 4 kind of phases of the journey of when it's viable and when it's not, now it's time to do it together and kind of show you a step-by-step of how I do it with the pollen and with that.
So the first thing you're going to do is wait for your Alocasia flower or inflow to be on that second stage where you see the spathe is erect. Then you're going to cut an opening on the side of the female area. Some people take out the entire structure. I personally believe that the berries and the inflow needs the humidity, it needs the temperature, and kind of like it's there for a reason in nature, and I believe it somewhat is necessary for it to be successful. So if you want to remove the entire thing so it's easier, go for it. If you just want to sort of recreate that kind of chamber effect, then you're going to have to cut a window through it. I've learned that trying to cut a square window is not really effective and usually end up just like cutting part of the berry flower inside. What I do now is kind of just follow along wherever it's popping out and protruding from like the stem, and then just kind of guide it down with a blade. I use a scalpel, but honestly, you can use any kind of sharp blade would work. And then you're going to select your pollen. Honestly, odds are you're just going to select whatever you have available. But if you're like me and you have many species that are consistently putting out inflows, that you might be able to collect them, I use centrifuge tubes. And then what I like to do is grab the pollen and mix it with some water, some DI water, until it creates a slurry. like a slushy. I guess that's a better term. Like it's going to create it thickens, it creates a slushy. If it's not looking a little sus, it's not quite ready, it needs more pollen. And then I just kind of grab a little bit of this glove, this paste, and then I kind of just very gently dab it on the actual pistil flower. So it's that little white top area. You don't want to rub it, you don't want to brush it, you kind of just want to dab it. You can also use dry pollen. It will stick if it's quite ready. Some of the alocasia species put out like this white substance that is really sticky. Other ones do not. So I find it to just mix it with a little water. It gives me the most consistency. And then you just want to touch every single one of those pistil flowers on both sides, on the side. And then you can do this maybe twice during the day at different times. So you kind of hit the window if you kind of want to make sure you hit at a certain time.
So if you did everything right, if you nailed the timing in your pollination efforts, if you just got the right temperature, because temperature is very important, that's why I don't recommend that you cut out everything and kind of expose it, because I believe that temperature plays a key role in the development of the pollen tube, and I believe that that is really important. So if you nail everything and you get it right, then you're going to start growing the berries. I told you that it changes the name into an infructescence, and that is exactly what happened to mine. You can see them right here. And honestly, I'm going to this is me burying the lead after you sat through this entire video. This is actually where the real work begins in getting your hybrid. Using the pollen to create a viable berry, that is like step one, difficult, but doable. Now, once you have the berry actually developing inside the inflorescence, now you need to wait between 3 to 8 9 months. Like straight up a full-blown human baby is how long it takes for some of these berries to mature. It will depend on the plant, it will depend on the conditions, and it will depend on, some extra factors. But you should expect this to last a long time. Like your dog will have 3 litters before these fruits are ripen. So you need to wait, and at this time, you need to maintain your plant in the most optimal condition. If you put it through shock, it's going to abort the infructescence. If you let it dry out and it just dries out, it will abort the infructescence. If you overwater it, it will abort the infructescence. So you basically need to keep it in this kind of perfect spot for the time being. Then if you have done this correctly for months and months and months and months and months, that is where I was with my hybrid, then they begin to change colors. They're going to change color from the green to a light yellow, to then it goes into an orange, and then it becomes a deep orange. And again, the final stage will depend on the species. Some of them turn fully like really bright red, other ones turn really bright orange. But the important thing is that you should know is they mature at different speeds. So at least in my experience, the one on the top mature quicker or earlier than the ones at the bottom of the infructescence. So it is not all of them at once. Something else is it kind of did not mature like overall everything. Sometimes I saw like one side of the berry would turn yellow first, and then orange, and then red, and then the other. So it was not like homogeneous change in color. It was kind of splotchy in my experience. So what happened with mine is it did create the berries, it did change the colors, but I think it changed the colors a little too soon. So my actual hybrid was basically lasted 3 months. Unfortunately, I don't know, like the reason that it changed colors, potentially it was because the plant was put through shock. So if you saw my other video of me melting my Alocasia collection over the summer because I put it into like acid water, that really affected my collection and it sent them into like a spiral. So this plant in particular went through that when it had the infructescence, and after that happened, it began to ripen. So I don't know if it ripened because it was put through that or if the time was right. But the point is, this one lasted 3 months, which was a lot less than expected. The only way and the only reason I kind of knew that that plant was kind of moving on from that infructescence is because it started to put out this leaf. It had 2 other leaves, as you see in the footage, and those leaves were there for like ever. Those leaves were there almost a year, over a year. Like definitely, once it put out that infructescence, it basically stopped growing. It didn't do anything until they started to change colors, and then it began to push out this leaf. And this leaf is the biggest one that it has ever put out yet. And now is actively pushing, as you can see here, a second growth point and a second leaf that I kind of just broke off a little bit. Oops. It is putting out a new growth. But eventually your infructescence will fully ripen and you can begin to pluck them. Again, I recommend you pluck them at different times. But here is where the journey with my hybrid unfortunately kind of ends. That is also me bearing the lead. Yes, getting the actual inflorescence to become an infructescence is difficult. Yes, getting them to stay happy and stable and be able to survive, for 5 to 8 months is difficult. But in the end, this is the part where most of us fail that make it this far. So you will have your berries, and I put them in a paper towel. I kind of very gently, very delicately got rid of all of the pulp that was around it, the fleshy, meaty kind of thing, because I knew that would create mold and just be like no good. So I cleaned that out, and then I'm gonna tell you what I personally did. But since mine failed and ended up growing mold and kind of not becoming viable, I'm going to sort of tell you what happened, what I did, what I would do different, and then maybe you can give it a shot. And if you get to the same point, please comment down below what you found to be your success. So what I did with them was I took them out of the flesh, and then I put them in water for a few hours because I wanted to make sure that it soaked up kind of and loosen the surrounding. So one of them actually fell to the bottom of the cup. Like, it was floating at first, and then when I got back, it was like at the bottom. And I was like, well, that means it's viable, right? Like, if it's not floating, it's viable. So I put it into a container with perlite and stratum because that is what I consider to be the best kind of mix. I literally put in the same container as I do for my corms, and that was same thing that I do for my tissue culture. So that was the first one. The second one was the smallest berry that I had. That one was really brown, and when I put it in water, it stayed floating. So that one was, I was like, okay, maybe this will not be viable because it like stayed kind of floating and it has this harder shell. And then finally, the last one was the one that I had the most hopes for. It was the one that was the long one. I actually kind of hoped it would have two seeds in there so I could like pull it off. And that one I put in the water and it also sank. But then something weird happened in which maybe this was my mess up. Maybe I should not have put it in water. I should have just put it directly into the ground at this point. It had this white tissue coming out of it. And I think me handling the seed with tweezers while this tissue was out to put it into the substrate maybe killed it. I don't know if it was, whenever you open a fruit that is kind of like really ripe, it comes out with the plant already kind of sprouting itself, and you're like, oh, it's already kind of growing a new avocado or growing a new mango or whatever. This kind of felt like that. I was like, oh shit, it looks like it's already kind of coming out. I was like, I should just put it into substrate. So I put all three of them into the same substrate. 3 berries, unfortunately, is not enough numbers. Like, the odds are just not on your favor. Like, if that thing should be creating 20 and you're getting 3, those odds are already low. And then 2, I did not try different substrates. Again, with 3 berries, it's kind of difficult because you could not know what is the kind of thing that is helping or not helping. So I wanted to do them all in the same. But hindsight, I should have probably done like maybe perlite and sphagnum. My fear with that is that sphagnum tends to get really moldy as well, and it tends to hold a lot of moisture. But I guess, I failed with perlite and fluval, so would have been worth a shot. But if there's one thing I am, is persistent, and I will continue. So I do have a couple other fun hybrids. I don't want to honestly bring them. I already brought the Aslanii. The one is here in case. I have been known to drop my plants and fuck them up just for the video. So let me just show you some footage. I have 2 current ones that I have like the biggest hopes on. So my Longiloba X Corazon fail. That was really exciting because it was a variegated Longiloba. So I was hoping a variegated hybrid or something along those lines would have been cool. And then the other one is going to be the Aslanii. And as I mentioned, I think earlier, that is a mellow hybrid. Basically, I was able to get mellow from the inflow that came out of the little plants. I collected it because I was like, you're not doing well. And then I put it on that Aslanii. I just thought it would be an interesting mix. Again, Aslania is infamous, at least for me, for faking me out for a couple months and then being like, psych, I am going to abort everything. So who knows? Potentially right now, I'm seeing a couple of the berries are turning yellow. That is kind of how you know they're starting to fail, is because they shrivel, they turn yellow, they kind of just like, yeah, like a raisin kind of lose all of its moisture. And then the ones that are correct, the ones that fertilized, get huge. And I'm seeing a few that are starting to get bigger in my Aslania mellow. So fingers crossed. But again, after this experience, like the first step, I got it down. Like I will keep just mixing my plants and getting some fruit, keeping it alive. Like I feel confident I can keep my plants alive. I can keep them healthy. I can keep them happy. I can keep them growing. But that last step is going to be the killer, because everything's out of our control at that point. That point is going to be the substrate, is going to be your seed, is going to be that. Maybe in the future, what I would recommend and what I would do is use a heating mat. So similar to maybe corms, they do better if they're warm and in a heating mat. Potentially the seeds will do better and sprout, be more viable if they're in a warmer kind of spot. I don't know. This is just me kind of like throwing it out there and guessing. I would need a lot of different seeds to be able to test out something that would feel confident being like one or the other. A quick little section that I wanted to add here is cutting or not cutting your inflorescence or flowers. So this is going to be really dependent on the plant. So if you have a plant like this that is actively pushing out new growth while simultaneously pushing out the inflows, to me that means your plant is good. Like you do not need to cut them because they're going to go through the cycle, right? The energy has been spent. If you have multiple, you can play with it, you can collect, you can try them out. If you don't like them, you don't want to deal with the smell, you have zero interest in creating a hybrid, you can 100% just cut them off. It will not damage, quotation, like I need an asterisk at this part. It will not damage in itself, like you're taking away the nutrients. Like I don't think it's that important for it to like reabsorb the nutrients from a failed inflow. Like no, just put some in the water and it'll be just fine. But what you will do is potentially cause shock. So what I've noticed is whenever the flower is coming out and it's still kind of inside its little kind of containment that they come in, like a bunch of them come in this kind of, I don't know what it's called, like film over it. If you cut them at that stage, the plant will go into shock and many times stop growing. It's almost like it had its mind set on starting the inflows and you cut it off and now it's like, now what do I do? So what I've learned is that I need to let that part kind of come out, like let the inflow come out a little bit more. And most of all, I try to look for new growth. I need to look for a new growth of the leaf. I need to see a new leaf sticking up. I need to see the next inflorescence if it's like a multiple kind of like set of journey. What I definitely do not want to do is just cut the inflow and leave it like completely bare with nothing, because that to me has been like straight to getting your Alocasia to bolt, which is what I consider whenever it just creates a kind of like concave, dry middle with zero new growth for a long, long time. And trust me, your plant can stay like that for years. So you don't want to do that. If you see them coming, congratulations, you got your plant to an adult mature size. Great achievement. Now I recommend that you stay on top of it with nutrients and you stay on top of it with light, and yeah, you should be just fine. So now that we had a step-by-step tutorial, I wanted to talk about some interesting observations that I have personally made on my plants, some of the reasonings and why I think it happens, and then I would love, 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 love if you could share with me if you have similar experiences, what you think could be the cause of it. And yeah, I just want to learn. Like we don't have enough people doing this research. Something that I want to start with is we're kind of pioneering a lot of these kind of things. Like something that I liked is all of these plants produce pollen. And I wanted to look at them under the microscope. So here you can see specific plants, what their pollen looks like under the microscope. So after you have tried to fertilize and you put the pollen into the female area. So let's assume like this is where the female area, you cut up a little hole right here, you put your pollen, everything is fine. Something that I've noticed is that many times the plants will instantly kind of go and then turn this upside down. it kind of like it bends itself in, yeah, upside down. And at first I was like, oh no, something wrong is happening. Let me like make it stand up or attach it to the side or bend it so with a petiole so it's not like flipped. But then I noticed that it was happening with some plants and not with others. And I noticed that whenever it seems to get further along in the whole kind of like creating a hybrid and maybe you successfully fertilize one of those flowers, I've noticed that it's when it flips. So I don't know why it does that. It kind of just, yeah, it kind of bends to the side. Some of the ideas and thoughts of like, why would a plant be doing this is one, I think maybe if you are in the rainforest and it's raining a lot, like potentially water or liquid could just accumulate inside the female area. And as a way of preventing it, I feel like maybe it turns around to sort of tip that over or make sure it doesn't get filled because it doesn't have a lid, kind of like Nepenthes or other one of those type of like Bornean species. So that was my first thought. The second one is that it could be similar to like a Dutcherman's pipe, in which it traps inside the flower its pollinator. In this case, I believe is flies and like little beetles and probably ants. So whenever it closes that interstice, basically everything is trapped in there. So I wasn't really sure if that is a mechanism to like trap them, to make sure the touches start to escape through everything. And then whenever you flip upside down, you force them to come out through that space and that area that has all the pollen accumulated in it. So you would basically guarantee that the bug is covered in your pollen when it has to leave that area. So those were like the two main kind of thought reasons behind it. Whichever, if you have observed something similar, if you have learned or you've read something, please share down below. Many times a lot of the research to answer these types of questions never make it to our side of the biology horticulture, because we are on the ornamental end, which is considered obviously not as important as the crop side. So a lot of the research, the money and the science goes towards crops. All of us with the ornamental, we're left to kind of like figure it out on our own. So I'm not sure what would the real cause of that. I'm not really sure how you would do in tests to try to figure it out, but that is just something that I have observed. The other one is going to be the arrangement of the berries. So all the berries look very different, or like the flowers, the pistillate flowers look very different depending on the arrangement. So for example, Maharani has almost like a wall of tiny little ones everywhere. And that is really similar to how what it looks like in the mellow. But the Reginula looks a little different. For example, Aslanii, as I showed, it already has like almost like this perfectly formed berries out of the gate. Other ones will have 2 rows or 3 rows. Other ones are gonna have 5 rows. The same plant might put out they will put out multiple inflorescences, but the same plant with multiple inflorescences, like the flowers will look different. Like some of them will be bigger and have more rows, other ones will look smaller and have less. So I think that is just normal variation kind of like in this, but it's something interesting to note is there's a huge variation in the arrangement and the shape of those flowers. The other one is going to be the color of the spadix. Some of them have a bright orange, almost pinkish color. Other ones just have the white one. So that is an interesting sort of distinction. And within that, why I think it's interesting is sort of like the way it attracts the pollinators with like smell, but also temperature. So I believe they used to think that the appendix is what was producing the smell. I don't believe that to be the case. I believe it's probably coming from the actual pistillate flowers, because they have this gooey white substance that comes out of there whenever you put in the pollen at the right time. Is super sticky, and that smells exactly like whatever it's out into the air. So I believe the upper part, the appendix, is probably more for temperature. It's probably just getting hotter and indicating like for those bugs that can actually detect that slight change to indicate whenever it's like the most optimal time to come to the inflow. But the interesting part is the male area, which is the one that is producing the pollen, is going to be above the sterile interstices with the appendix, because all the pollen kind of falls on the lid and then it kind of stays there. But that is not the case for all of them. I noticed that there's certain ones that they have the male area inside the female area, and then it kind of closes at a weird angle. Some of them produce a lot of pollen and it goes inside. So whenever you open the space in that lower area, it has a thick part full of like pollen from the same plant. So that whole angle of them closing to prevent self-pollination and also flipping upside down to prevent that could potentially falter because some of these plants actively drop the pollen inside. Potentially some of them are able of self-pollinating while other ones are not. Again, for everything that has to do with this small jewel alocasias, we just don't have enough kind of information for that. So there you have it. After this, you should be an expert in the anatomy of Alocasia inflorescence. You should be an expert in how to try to get your hybrid and make them like fertilize that inflow. And you should hopefully have some ideas and some tips on how you can make your seed succeed and sprout. Comment down below if you have any hybrids in the making. Is this video helped you? This is everything that I wish I would have known whenever I started my journey and I wanted to learn more about inflow.